So my name's Bill Matsui. I'm here actually in Austin. I've been here for about a year and a half. And I came from Johns Hopkins. I moved from Baltimore to Austin. And if you want to know why, come and ask, and I can tell you the whole convoluted story about how I got here. But I think that having, one of the things when I was, after I got here, was an opportunity to do a patient education series for myeloma. And for me, this was really important because, you know, myeloma is a disease where I take care of, the most patients I take care of have myeloma. And I think that it's really like one of the points about coming here to Austin was to try and develop programs that were centered around specific diseases. So I take care of patients with all blood cancers, but most of them have been myeloma. So it's really an opportunity to sort of not only establish a program at the University of Texas, but to really interface with all of the people who are here in Austin who take care of myeloma patients, and then see if we can really sort of coordinate the care of patients along the way. So I think a really important part of what the myeloma crowd is doing is really this idea that, you know, you should have some empowerment about your own disease. So this has been a thing where a lot of people will come and they'll say, well, what can I do? What can I do about my disease, and how can I help and try to figure things out? And I think this is actually a really great way to do that, because it's something where if you look at many different cancers, there's not anything like this that's been available. So it's really something that's cutting edge, not only for myeloma, it's a cutting edge thing for all of cancer, to really get patients involved in their care and to use really information from patients to help try to figure things out. So I think that's really sort of a modern way to do things. It's different from what we did in the past, where I was at a university, and the only patients you really got information to do trials or to understand things were really just either your own experience or patients you had to enroll in a trial, and it really limited the number of patients you had to study. So today I have like two jobs. One is to talk about sort of this idea of risk, and then to bring in the idea of how do the genetics of myeloma influence that risk. So we're going to talk about those two things. So the first is we're going to do risk assessment. So I think that when most people get diagnosed, when I have a patient who comes with myeloma, I usually talk to them about a couple of things. One is I try to explain to them what the disease actually is, and then the other is that how do we think about the treatment? And part of the way we think about the treatment is derived by this idea of risk. And so myeloma, can have a wide variety of ways it presents, like we heard that from the video. And then the other is that it has a very, very wide range of what happens to patients once they get diagnosed. So there are some patients who the disease can act much more aggressively, can grow very rapidly, and those patients can get sick very, very quickly. And then there are others who can have slower growing disease. And so if we're able to figure out who has more aggressive disease, maybe we should treat those patients much more aggressively, right? And if we think you're going to have slower growing disease, then maybe what we should do is, in some cases, so the extreme of this might be something like smoldering myeloma, well, we might not treat you at all. We might just watch, right? Or if you have certain kinds of myeloma, maybe perhaps what we should do is not treat you with sort of a nuclear bomb of the treatment, but think about using things that are much more tolerable. So when we think about risk assessment, we think about a couple of things. Like, what are the things we're trying to figure out? Well, one is that we're trying to figure out, like, if we have a patient, we have certain parameters, we know about them, what is the probability, or what's the chance that if we treat them that they'll go into remission? What's the chance that, what's the period of time that they might stay in a remission before they relapse? And then the other is from the time of diagnosis, how long are patients going to live? So really, the two parameters we like to look at are really how long are people going to stay sort of in a stable state or even a remission, and then how long are patients going to live overall? right? So those are the two things we use when we're assessing risk. So the one thing that is super important that I try to explain to patients, and I haven't really figured out the best way to explain it, is that when we do risk assessment, right, it's a probability. It's a chance that something is going to happen, right? So like if we say, okay, well, if we go and we drive from here to Dallas, okay, there's some risk that we'll get into a car accident. There's some risk that we'll like die in that car accident, but it doesn't mean that that's going to happen to us, right? So risk itself is a probability, right? Risk itself is not saying that if you have low risk of the disease, this is what's going to happen to you. If you have high risk disease, this is what's going to happen to you. It is all based upon a probability, okay? So it doesn't mean that's what's going to happen to you. So that's the really important message. So if we look at risk assessment, really, we try to divide people into sort of high risk, low risk, or even into three groups where you have intermediate risk. And so what you can see on the slide is that there is, let's see if I can get this to work. So basically what we have is sort of the survival of patients. So we might see some of these graphs today. So this is a probability or the percentage of people that are alive. So at the beginning, everyone's alive, right? So it's 100%. And then as we go out in time, there are patients who unfortunately pass away. And so there are different rates at which that happen. So that's how we sort of divide people out. So patients who have lower risk disease might be up here, right? And then those with higher risk disease might be down here. So when we think about risk, how do we think about it? How do we sort of piece it together to sort of say, look, we think you're in one of these types of situations? So there are really three things. One thing I think about is the patient themselves. The other is really the stage of myeloma, something we do with all patients. And then the other are tumor factors, and those are primarily the genetics. So I was going to talk about those three things very briefly. So when I first meet patients, the things I immediately think about are, one is what's the age of the patient, because that can dictate what we might offer the patient as treatment. And then the other thing is this idea of performance status. So performance status is really something that's been used in clinical trials where you say, okay, if we have patients, we want to have a sort of a very general gauge of their health. So the way we do measure that is like, how active are you? How much time do you spend in bed? You know, how mobile are you? All of these things. So there are a number of different ways. There's a scale from 0 to 100. There's another scale that goes from 0 to 5. And so really what we try to do is try to figure out along that scale, it's really just an estimate where people are. But if patients are sicker, then it's harder for them to tolerate some therapies than if they're not as sick, right? So that comes into the play of what we're going to do. You know, when you get diagnosed, you know, you think that people draw like a gallon of blood from you, and so we do all of this testing. And so a lot of the test results we get not only help us diagnose the disease, but help us figure out sort of where the disease is affecting your body. And some of those parameters can actually help us establish this idea of risk as well. Okay, so we're going to do some blood tests and we're going to figure that out. So really in the early days when people were looking at, you know, at stages of myeloma, so like if you talk to people, let's say with, I think like with breast cancer or colon cancer, like solid tumors, they'll say, I was stage 1 or I was stage 2. I was stage 3, and that is sort of the extent of spread of the cancers in those solid tumor patients. So if someone has stage 3 breast cancer, they not only have a breast cancer in their breast, they also have breast cancer that goes to the lymph node, okay? If they're stage 4, then it's spread further away. So obviously, the higher the stage, sort of the more extensive the disease. In myeloma, we use stages as well, but the stages are much more sort of fluid, because myeloma is a disease of the blood, and the blood goes everywhere, so it's not like it's localized to one place and goes in a predicted fashion like a solid tumor. So when we're thinking about stage, there are a number of different ways to do it. Initially, when staging was done, it was all based upon what your M protein was and how many plasma cells you had. And so over time, that was not really the best way to establish it. And so there was this new staging system that came out called the International Staging System. It's based upon 2 values in your blood. One is this thing called the beta-2 microglobulin. The other is a protein called albumin. And so we just measure those two things, and then we can sort of assess what stage you are, and then these are the outcomes for those patients at each stage. Right, so this was done with a large number of patients. So that's one way we can stage. Does it really matter what stage you are? Like, to me, it's not that important personally where you are, okay? Like, I think that stage 1 patients need treatment like stage 3 patients do. I don't necessarily change how I do the treatment myself based upon the staging so much. The other is that one thing we do is we have this focus on genetics. And so just like every other cancer, there are genetic changes in myeloma. So what I thought I would do is talk about a couple of the ones that we typically use, and you might get these results from your doctor. And one thing to do is try to understand what we're talking about. So one is to think about like chromosomes. How do chromosomes change in patients in their myelomas? And so we have 46 chromosomes in each cell, right? They're pairs, and so you can sort of do this special test and then lay them all out. And then what you can find is that you have 22 pairs, so there's a doublet for each one, and then you have an X chromosome and or a Y chromosome, 2 X's or an XY. And so chromosomes themselves are arranged where there's this thing in the middle, like this hub called a centromere, and then there are a short arm, which is the P arm, or there's a long arm, which is the Q arm. So what we can do is we can, originally when people thought about genetic changes in cancer, this is what we could do. We could see whether chromosomes looked funky or not. So this was done way, way, way in the past 60 years ago. So what can we do in myeloma? So in myeloma, we can find sort of two sort of distinctive patterns of chromosomal changes. So one is called hyperdiploid, where you just have extra chromosomes. So you have extra chromosomes, and typically you'll have, instead of 2 chromosomes of each pair, you'll have an extra 3, an extra chromosome 5, a 9, an 11. So it's, for whatever reason, it's these odd number chromosomes. So you can have extra copies of that. That happens in about half the patients. And then the other ones are these things called chromosomal translocations, where you have one chromosome sort of breaking apart and another chromosome breaking apart, and then they come together in an abnormal way. So you're making sort of a new fusion chromosome. So this happens in about half the patients, and what we see translocations that involve, it's one chromosome, chromosome 14, and that's the chromosome where the gene is that makes the M protein. So that is overactive, and so you can have chromosome 11, 14, 414, all of these combinations, and so this is what we see in about half the patients. And then the other chromosomal changes you can see are either part of the chromosome is gone or part of the chromosome is actually amplified. So those would be gains or losses. So these are the 3 general types of chromosomal changes you might see. And so when we look at a myeloma patient, for the example, so in this patient we have all the chromosomes lined up, and in this person they have an extra copy of chromosome 3, chromosome 5, chromosome 9, 2 extra copies of chromosome 11. So this is a person that has these extra copies, okay? So this is sort of what we would read out. This is actually a test that is not that useful in across the board in myeloma patients. And the reason why I say that is that to get this, you have to get the myeloma cells to divide and then freeze them in a place where the chromosomes are ready to split apart. And myeloma cells themselves, especially when you have them in the lab, don't really like to grow. So then you can never get to the point where they can divide, so you can never get this picture, right? So when people do cytogenetics, what's called classical cytogenetics, which is this, oftentimes the test is going to be negative. So it'll look like you have a normal set of chromosomes, but you're getting that set probably from some other blood cell, like a normal blood cell. You're not getting it from the myeloma, okay? So this test is useful in a portion of patients, but not in all patients. And so what else can we do? So the other test that people oftentimes will get is something called FISH testing. So what is FISH testing? So FISH testing is, if we have a chromosome here, what we can do is find we can make sort of like an artificial gene type thing. And what we'll do is we'll put a little beacon on it, and that piece of DNA will now go and find its partner in the chromosome. And so what you'll have is you'll have the chromosome actually now have a piece of it lit up. Okay? So it allows us, even if the cells are not dividing, so we can get that nice picture of all the chromosomes lined up, we can still figure out, are there changes in the chromosome even if the cells are not going through cell division. So this is sort of the standard way now that we judge myeloma genetics, right? So when you get a bone marrow done, it may not be every time you do it, but oftentimes in the first time you get diagnosed and you have a bone marrow done, this is the test that people typically do. So this is what FISH testing is. And the reason why I bring this up is because a lot of people say, well, I don't understand. I don't understand, like there's this test and this test and this test. So this is really just one way of looking at chromosomes. Okay? It's not looking at specific genes. So if we go through and we look at, how does this help us with risk, there are certain cases, like if you have extra chromosomes, you're typically a standard risk or low risk. If you have extra copies of chromosome, the long arm of chromosome 1, or if you are missing parts of chromosome 13, that might be intermediate. And then there are these high-risk cases where you either have less than the normal number of chromosomes, or things like where you're missing specific parts of a chromosome like 17. So this is one way that helps us sort of figure out where you are on the spectrum of risk, right? But it doesn't necessarily, like I said, if you have a high-risk case of a high-risk chromosomal abnormality, it does not mean that that exactly is what's going to happen to you, right? It's a probability thing. It's not, it's not a fate. So people have done sort of this staging, and then the realization was that you could use these genetics to help you. And so in the end, people combine these two things, and so you have this new sort of revised staging system. So stage 1 is the old stage 1, but then you don't have any high-risk things. And then the other thing they added in was this one blood test called an LDH. So stage 3, which is the highest stage, is you're the old stage 3, but then you have a high-risk thing, or you have a high LDH, and then the thing in the middle, RS, like stage 2, is like everything else that doesn't fit in 1 and 3. So if we look at that and then we compare sort of how do patients do, if you're a stage 1 patient, in this study, right, at 5 years, 82% of those patients were still alive. Okay? If you were high risk, then at the end of 5 years, 40% of the patients were still alive. So what we're looking at here is either the length of overall survival, or if the patients got treated, then how long were they still in a remission or a stable state before they progressed. So at 5 years, it was 55%, and then for stage 3, it was 24%, okay? So what one thing that's important to say is that it's not like, none of these are like 0% or 100%, right? They're all variability. So what about beyond chromosomes? We talked about these chromosome changes, right? Those are sort of the entity at which all the DNA is wrapped up in. What about the genes themselves? So if we think about like the chromosome, so if this is sort of the chromosome, we have 46 of these, they're all sort of a conglomerate of DNA. And so the DNA is what encodes the genes, right? So all the genes, we have about 20,000 encoded genes in each set of DNA, so in us. And so there have been studies that have looked at what are mutations that happen in genes in myeloma. And if we look here, there are some genes that are more commonly mutated than others. So KRAS and NRAS are two. So that's about a quarter of patients or a fifth of patients. And then there are a lot of genes that are less than 10%. So one thing that's different about myeloma than some other cancers, and so like a good example would be like, let's say pancreatic cancer. So in pancreatic cancer, if we did this test, just about 100% of patients would have a KRAS mutation. So they all have this mutation. In myeloma, there are a bunch of different genes that are mutated, but there's no one single gene, right, that is mutated in every single person. So there's not like sort of in pancreatic cancer, people say, oh, well, KRAS, if KRAS is mutated in all the cases, then KRAS must be important in sort of starting the cancer or having the cancer be initiated. We don't have sort of that kind of thing in myeloma. So myeloma starts from a bunch of different places. It's not like genetically sort of distinct like it is for a bunch of other diseases, a lot of leukemias, and for things like pancreatic cancer. But we can find mutations there. They're oftentimes there. So if we have this information, let's say we have the cytogenetic information, we have the FISH results, we have these gene mutations that we do in some patients. How does that help us? Well, it guides us, but it doesn't necessarily tell us exactly what to do, okay? So for examples of this might be if you had a deletion 17p, which is a higher risk cytogenetic abnormality, then one thing might be if you get a stem cell transplant, maybe we would give you 3 drugs instead of a single drug as maintenance. So oftentimes what we'll do is we'll give people Revlimid as a maintenance drug after a transplant, a stem cell transplant, and then, but if you have a deletion 17p, maybe we'll think about giving you both Revlimid and Velcade and dexamethasone as maintenance. The other is that if you're at high risk, maybe we think of doing something else besides an autologous stem cell transplant, and Dr. Ramakrishnan will touch on this a bit, but we might do a different kind of transplant. There's some thoughts that there are some chromosomal abnormalities that are where some drugs work better than others, and so there's studies being done with this one drug, venetoclax, and there's interesting things about this drug in myeloma patients, but it's thought that this drug works better in patients with this chromosomal abnormality than patients with other chromosomal abnormalities. But this drug is not yet approved for myeloma. There are specific genes that can be mutated, and so we talked about KRAS and NRAS mutations, and so there's some reports where inhibitors that inhibit the pathways where these genes are involved might be utilized. There are about 3% of patients will have mutations in this gene called BRAF. BRAF is oftentimes mutated in melanoma, and so there are actually inhibitors that are used in melanoma patients for patients with BRAF mutations. There are some thoughts about using BRAF inhibitors in myeloma, but that's also not approved. So it's really like what we're trying to do in the end, like the great thing to do in the end would be to be able to use this genetic information and say, look, you have these genes or these things that are mutated, so this is like the best set of drugs for you, right? So we're not close to that in myeloma. Well, I would sort of say we're not close to that really in any disease in cancer, but it's one of the goals that we have is to sort of say, for you, it's not based on your stage, right? It's not based upon how many myeloma cells you have. It's really based upon like the things that are molecularly wrong in the cells. So why don't we try to attack those things? So that's one of the goals of many of our groups. The other thing is that how do these things change over time? And so one of the things to look at is, if you have a patient, so this is a patient that was reported out by a colleague of ours, Jonathan Keats, where you have like a patient, they were diagnosed, they got, Revlimid and Dex, their M protein went down, started coming back up, so they got carfilzomib, went down, and et cetera, et cetera, et cetera. So this is a very sort of common scenario where you get a drug, you go into remission, comes back, you get another drug, and it goes up and down. So you can see here that the myeloma itself is inactive and then becomes active again, and it's influenced by all these treatments. So one question is, well, how do the genetics change over time? So this is an interesting story where if we look at that patient at diagnosis right here, it ends up being that if you look in this pie chart, I know it's hard to see, but there are like 1, 2, 3, 4 different things. There are actually 5 different things. So what this means is that in this patient, when they were diagnosed, they had actually 5 different genetic variants of their myeloma, okay? So they started out with 5, and there was one that was about 3/4 of all of their myeloma cells. So if we took 100 myeloma cells, 72 of them would be this, 11 would be orange, 10 would be green, et cetera, et cetera. So they got their first therapy, and then it switches a little bit, right? So the orange becomes more, the red becomes less, and then they get more, that's when they're in remission, they get more Lendex, and then when they relapse, now you have this green guy coming up, right? So there's a change there, and then you give another set of drugs, it's orange, orange, and then in the end, it ends up being purple, right? And this is the thing that was very, very aggressive. You could find this thing actually in here, the same genetic sort of clone, but it was very, very small. So in the end, the thing that was very small was the thing that actually, unfortunately, was the most lethal thing for this patient. So these things will change over time. And like I said, there's differences in response, and then in the end, the small one in the beginning became the bad one in the end. So how do we think about this? So if we have multiple different kinds of sort of genetic variants of your myeloma, and they each are sensitive to different drugs, then maybe what we need is to give like good combinations of drugs. So that's one of the thoughts of giving more than one drug. It ends up being that the cells can change so that the makeups over time, and so there are cases where, and this is something we bank on, is that even if you have a drug has stopped working, that that one that was sensitive may grow back later, and you might be able to use the same set of drugs again. And then the other is that these minor ones, these very small ones, may be things that are the most aggressive in the end, and we need to try to figure out, well, why are those things small in the beginning, and then what makes them big, and what makes them resistant to the drugs. And so that's sort of what a lot of our research has centered around. So in the end, the things about risk, there's many different things, and so like for me, a lot of it is you can look at things like the genetics, you can look at things like the stage. I think it's very important because what you're really trying to do here is figure out what the best way to treat a patient is, and so you got to look at the patient as well. That's equally as important as all the other stuff we do. And so the risk represents a probability. It's what's the chance of something happening, not the destiny. That's not what's going to happen. And then it can help us figure out what we should do, like what are our treatment goals. It's really important because some clinical trials are available for some sort of genetic abnormalities or some types of patients. This risk assessment we do in the beginning, it's not that useful when patients relapse. So I don't really go through and say, look, let's do a whole assessment again. And then what we're doing is there are a lot of efforts ongoing to use genetic information so we can match people up with different either clinical trials or different therapies, and so that's a major goal of a lot of what we're doing. So that's what I wanted to talk to you about today, and so if there are any questions or we can ask, I don't know what the timing is, but we can do that now, or we can do that, I'll be around all day, so feel free to just let me know. Okay. Okay, so we'll do that.
At the end of the morning.
Okay, sounds good.